In this one-year pandemic, scientists had immediately gotten to work to finding a vaccine for a recent killer. Remarkably, they eventually did, and this 3D model will explain their creation and how it works. Now, let me take you down to a world on the microbe scale. And here we are. In a detailed animation, this is SARS-CoV-2. In more general terms, the coronavirus. The virus responsible for the deaths of millions in the past year. Looking at it by itself, it appears to be a small and a harmless cell. But a replicated 100 million of these, not so much. You start by acknowledging the cell itself. We see that the surface of the cell is coated with spikes. It uses these spikes as leeway to enter in human cells. Under the spikes, we see a layer of lipid fat. Now remove that layer, and we begin to see the internals of the cell, consisting mainly of genetic material, specifically the RNA for the spike protein gene. Scientists then extracted the RNA, and the viral genome was later decoded, and eventually it was possible to deduce what RNA sequence codes for which protein, the spike protein, for example. So it was decided that the vaccine would need to contain the mRNA that our cells would then use to make proteins with. But mRNA is extremely fragile on its own, and it would be chopped to pieces by our natural enzymes if it were injected directly into our body. To protect the vaccine and also allow passage through the cell membrane, Pfizer and BioNTech wrapped the mRNA in small bubbles made of lipid nanoparticles. The lipid nanoparticles containing the RNA would then be delivered into the patient via syringe. After injection, the nanoparticles are absorbed by the body and taken up by an antigen-presenting cell through endocytosis. It then releases its RNA package. Next, the RNA is translated into viral proteins and is then cleaved into fragments through the proteasome. The fragments are further processed by the endoplasmic reticulum to make MHC, which is then transported to the cell membrane. Now, where the vaccine actually works. When the MHC1 spikes are presenting itself outside the vaccinated cell, one of three things generally occur. 1. The vaccinated cell would eventually die, and the debris containing many proteins and protein fragments would then be taken up by a type of immune cell called an antigen-presenting cell. 2. B cells that may bump into the coronavirus spikes on the surface of the vaccinated cell could lock onto the spike proteins. If these B cells are then activated by helper T cells, they will start to proliferate and pour out antibodies that target the spike protein, which then the antibodies would latch onto the spikes and mark the virus for destruction and prevent infection by blocking the spikes from attaching to other cells. 3. CD8-positive T cells, which is what we are going to do in this case. Also known as killer T cells, they bind to the MHC1, creating a memory of the viral antigen, and given the name killer T cells, they would then lead to attacking and destroying the infected cells as long as it's in their memory, which studies show would retain in their memory for decades. So after the administration of this vaccine, our immune system will be ready to save the day. So here's a small but detailed example to show us exactly how the immune system can protect us from this virus. So let's say hypothetically, after being vaccinated, your bodies come in contact with SARS-CoV-2. The antibodies that were originally created when you were first vaccinated would then recognize the virus cell as foreign and would essentially bind to the spikes since they are originally primed for them. So as I said, multiple antibodies would cover the spikes, which would then restrict the cell from entering a human cell, as now the coronavirus cell would not be able to fit through the ACE2 receptor, which would have originally granted them access to the inside of the cell had they not have been vaccinated. So, all in all, I personally feel, once vaccinated, COVID would not be something for all of us to worry about for a very long time.